Please welcome Bo Norrman, Chalmers University of Technology.
Hello, my name is Bo Norrman and I work as an innovation advisor at Chalmers University of Technology in Gothenburg, Sweden. At the Innovation Office, we support novel ideas from researchers by validating their IP, market awareness and technology. I have nominated Elypta, a company developing cancer diagnostic methods based on systems biology. The concept of using changes in cellular metabolic pathways as a diagnostic tool is a novel way of detecting the transformation to cancer cells. I've also known Francesco Gatto, the CSO and founder, for several years, and I've seen how his knowledge, business understanding as well as team formation has been essential for going from idea to a growing startup. Based on this, Elypta is a worthy contender to become a science startup breakthrough of the year 2022.
Breaking the wall to multi-cancer screening. Carl Bergman, Elypta.
Hi everyone. So cancer is on its way to becoming the leading cause of death in the world, and our vision at Elypta is simply that it shouldn't be a cause of death at all. 3 out of 4 die when cancer is diagnosed in stage 4. But when it's found in the early stages, mortality drops significantly, and so does the treatment costs. So to detect cancer earlier, we need to screen healthy people before they have symptoms. And today we do this for breast, colorectal, cervical cancer. But if we're ever to find this long tail of other cancer types, we need what we call a multi-cancer screening test. And today, there's really only one test on the market, and this is only in the US. And while it has few false positives, we estimate it detects only 10 to 15% of these early stage tumors in a healthy population. And the price is too high at $950. So what we need to break the walls to multi-cancer screening is to double this detection rate and slash the price by 70%. To do this, we must go beyond DNA and proteins and explore human metabolism. And our founders asked the questions, which metabolites change the most in cancer? And it turns out to be something called glycosaminoglycans. 17 of these metabolites are found in blood and urine, and Elypta spent the last 5 years developing new laboratory assay kits to measure them in a reliable and scalable way while training algorithms to detect the patterns that are specific to cancer. In a clinical context, the process is simple. We collect 2 drops of blood and urine from a healthy person. This is sent to an existing diagnostic lab. They use Elypta's kits to extract the GAGome and then measure it on a mass spectrometer. The data is uploaded to our cloud-based software, which computes a score indicative of cancer or not. And the whole process takes just 2 days. With 60 instruments, we could screen 1 million people. We used this approach in a multi-cancer proof of concept study, including 1,000 people and 14 cancer types, and presented the results at ASCO last year. And you see here on the y-axis is our GAG score. Purple dots are healthy control group. And then you see the clear rise in GAG score already for stage 1 patients. Overall, we found 35% of the cancers here with 1% false positives. And importantly, we detected all the 14 cancer types. So we wanted to validate this in the true real-world screening-like population of people who are presumed healthy. And we did, and we found 21% of stage 1 cancers up to 18 months before they were eventually diagnosed. So this is a breakthrough. And to get this test to market, we're now finalizing in vitro diagnostic regulatory standard kit design and scaling up manufacturing together with our partner Merck. Then follows clinical validation in 2 settings. Multi-cancer screening, of course, but also and here we already collected some 5 of the 10,000 samples we expect to need to get to market. But also a more narrow setting, detecting relapses in kidney cancer patients. And here we have a pivotal study running on 26 of the world's best cancer centers. The multi-cancer screening market is estimated to above 25 billion euros in value. And we'll first address the private pay segment. perhaps at a price point of 600 euros. Our cost, however, is just around 100. So we can grow this profitably, and the intent is to lower the price and address public screening markets in Europe, US, and globally. We carefully built a team of with both deep expertise and an innovative mindset. And with a 20 million euro Series A that we closed in June, we now have what we need to get to market. So what we have here is a breakthrough in sensitivity to stage 1 cancers and also a breakthrough in cost. And this can make the technology accessible to a broad population. We did modeling on what impact this might have, right? And we concluded that if we're able to shift the diagnosis using this type of performance, if used fully in the Europe and US, we're talking 175,000 extra survivors per year. So there's a lot of lives sort of in the balance here if we succeed. But to do this, we need to work with healthcare providers, we need to work with payers, we need to work with also pharma companies to explore the full potential of early detection and work towards realizing our vision. Thank you.
Thank you. Oh, wow, we already have a question, so we can start right away with the 4 minutes, and we start with Martin. Thank you.
Do you have any idea about your false positives? I mean, how many did you actually detect which weren't stage 1 cancer?
So in the proof of concept study just shown, we had 99% specificity, so around 1% false positives. But in our final test, we actually set this first. So we're designing the test to have 99.5% specificity, plus minus 0.25%. And we've set this level based on having a level of false positive that's acceptable to the healthcare system.
Next question here in the second row. Thank you.
Yeah, thank you. It's absolutely fascinating to be able to screen for 14 cancers simultaneously, but just out of curiosity, are there certain rare types of tumor, because we know how smart cancer is, that are not reliant on those metabolites stemming from the same substrate which your kits cannot detect still?
It's very possible, but if I was to guess, we've done many different studies, and we've never been able to find a cancer signal. So we think that we are detecting a signal that is more fundamental, something that changes early on when a cancer starts to grow. And that's what we're capturing. But we need to do much larger studies to prove this, of course.
Thank you. Geneviève, please.
Some cancers start very small and your immune system takes care of it. So are you interfering in that zone, or you're waiting for it to become really stage 1? And if I may, a second question, how do you differentiate once you know this person has cancer? How do you differentiate among the 14 types that you've tested for?
Both critical questions. So the first one, on the mechanism, we don't know for sure, but not all cancers in a very early stage don't have a significant immune response always, right? And still we detect them. So we think that the mechanism can involve partly the immune response, but also a physiological response of changing the biosynthesis of these glycosaminoglycans in the tumor's quest to try and affect the extracellular matrix so that it can grow. This is our hypothesis, but we need to do more work. The second question, when it comes to what happens when you have a positive test, this is the biggest challenge, and something that also involves the healthcare system. We've trained models in the study just shown where we try to see if we could provide practical guidance, like should you do an upper body CT to find lung and head and neck, or perhaps in the lower abdomen and find urinary cancers or something like that. And we had 90% accuracy to separating between those two. So that gives the doctor a guidance. When we split them by type, we had 74% accuracy to telling you the type. But what healthcare providers tell us is that they want to do this workup themselves anyway. They say, tell us if this patient has a cancer, we'll find it. That's our job.
Next is Stefan. Sorry. Yeah.
Can you talk about the defensibility of your research? I mean, how difficult, how long will it take for someone else to be on the heels of the GAGome?
We expect many to enter. We have our first US patent approved for kidney cancer, and we have many other pending across 18 cancer indications. But we also made sure to keep trade secrets around our assays. We're not disclosing the reagents, what we're doing, and the algorithms, and then the ever-growing database, now more than 100,000 data points on this. So, yeah, we want to be fast.
Okay, I'm sorry, we won't have it. I mean, we have, ask your question, yes, no question, let's go. 13 minutes.
It's too long.
No? Oh, okay. Well, then I'm afraid in 8 seconds we won't be able to make it. Thank you very much.
Thank you, everyone.